Okay, good morning or good afternoon, everyone. So today we are going to wrap up our discussion of the induced response component of our innate immune system. And as a reminder, this week you will have your normal weekly quiz due Thursday night by 11:55, and that quiz will cover Chapter 2 material as well as Chapter 3 material. So you can refer to the schedule that's on La Lima to kind of know what chapters or what lectures that corresponds to. Okay, so I encourage you to get into this routine of every week expecting the weekly quiz and knowing to look at the schedule file available in La Lima to let you know what you should be reviewing. Okay, so let's wrap up our discussion about the induced response. So a quick recap, we said that our innate immune system, the part of our immune system that comes kind of pre-programmed, right? It's in our genes to make certain proteins, certain cells that behave a certain way as part of our sort of built-in, quick and ready to act immune system that is modest to maintain, but very effective against everyday threats, everyday pathogens. We said that our innate side has two parts to it, two components to it. Our immediate response, which are the proteins like complement protein C3 and macrophages that are already there flowing through our blood or in our tissues, and those very quickly, right away, go to work opsonizing and phagocytosing bacteria and viruses. But if the number of pathogens or the nature of the pathogen enables it to kind of escape the immediate effector mechanisms of our innate immune system, then in the background, cells like our macrophages have been calling for help, right? Calling for backup. And that backup then is our induced response. So it still involves proteins and cellular components that are pre-programmed in our genome. It's just up until that point, our immune system hasn't really mobilized them because the hope is that our immediate response is enough to take care of things, because that's kind of the least expensive way to deal with a problem. Okay. So last time we looked at the recruitment of things like neutrophils as part of our induced response. So as cytokine levels build up from macrophages continuously encountering pathogen and sending out more and more cytokine messages, right at a certain point, you really begin to get more recruitment of things like neutrophils coming to help out. So let's finish our discussion with some other examples of induced responses. So this is a nice summary slide. It puts together some of the slides that we've talked about before, helps you distinguish the immediate innate response, which takes place from the moment that your tissues are compromised. So you get a cut or you get poked or something like that, and bacteria gets into your tissues. Within the first few hours, right, hopefully your immediate reaction or your immediate response is good enough, and that would result in very minor tissue damage. And in fact, you wouldn't even think about the fact that you were infected, right? So often I said, it's like, it's like you get a paper cut or you get poked by a pin or something like that. Chances are by the end of the day, you've forgotten about it and you don't even notice, right? However, if need be, your innate immune system can carry out the induced response. That is to say that as macrophages continuously are battling the bacteria, and it goes from just a few hours now beyond to maybe a few days, right, the induced response really begins to be involved. And that includes things like the neutrophils that are being released by your bone marrow coming out to help fight at the site of infection. And we learned last time that neutrophils are very effective phagocytic cells, but at the cost of them being sort of self-destructive, right? They can eat things a lot better than macrophages, but they can only eat a few bacteria before they've messed themselves up by changing their metabolism to the point where they can no longer survive. But they have that trick where they can explode and release the sticky contents of the neutrophil all over the place, including all the sticky chromatin that's carrying with it all those nasty enzymes that had been producing inside the cell. And that results to sort of different types of signs of an induced immune response that we're familiar with, right? The paper cut, or the cut that doesn't just go away after a day, but like 3 days later, you're looking at it and you're like, ooh, right? It's kind of reddish, it's firm, it's tender to the touch because of inflammation taking place. And sometimes also the involvement of very unpleasant things like pus, right? Which is very much evidence of the recruitment of neutrophils going to work. So hopefully that combination, though, with the induced response after a few days, then you've beaten the infection and you have some minor tissue damage, right? So that's why you might end up with like a little scar or something like that, right? I received a couple questions from your classmates asking about, this idea of the induced response. It actually seems like you increase the damage to your own tissues through things like inflammation, through the activity of neutrophils kind of going nuts and exploding and sending all their digestive enzymes all over the place. Yes, right. Evolutionarily, we have evolved to accept the fact that it's okay to cause more damage at the site of the tissue if it means that's what it takes to save our life, right? So it's basically the cost or the price of mounting a good induced response at the site of an infection is that you might end up with some long-term tissue damage there. Okay, but at least you're still alive, right? Because your immune system succeeded in beating the pathogen. This diagram on the bottom helps, hopefully helps you put this into context. An important thing to keep in mind, as with most biological, physiological things that happen inside of living cells, tissues, and organisms, things are very rarely a neat on and off switch between no and yes, on and off, right? Because we're talking about different types of biological components like proteins and cells, it's better to think of things as waves of activity, okay? Not just an on and off switch. The book, a little sloppy, I think, often refers to the change between the immediate response and the induced response as a switch, right? It uses it as you've now switched over to the induced response. But that's misleading because that's not true. This figure that I've drawn at the bottom helps illustrate that. Let us say that along the x-axis here is time, from a few hours to a few days, since time 0, which would be over here, which is the moment you get infected with something, like through a cut. Okay. And on the y-axis, I don't even have any units here because this is just a generic kind of response level. How active, how woken up is your immune system at this point? Right? So at first, when you have an infection, let's follow the blue dotted line, because the blue dotted line you can think of as the activity of things like macrophages, complement C3, right, going to work. So right away it goes to work, right? And maybe after a few hours, it's succeeded because the infection has been defeated by just the immediate players of your innate immune system. So as a result, since the bacteria is defeated and no longer there, your macrophage activity and complement activity goes down, right? And that's the end of infection. However, keep in mind that during those 4 hours, the macrophages are sending out cytokine messages. So what's happening is you do start getting the stirring or the slight bit of activity of your induced response beginning to turn on in the background, right, due to the cytokine levels getting higher and higher. But the point is that at this 4-hour mark, and keep in mind, 4 hours, I mean, that's just, we say 4 hours for convenience sake. It's not like, absolutely at 4 hours, this is when it happens. But after a few hours, if the infection was beaten by the innate immediate effectors, right, then the induced response also just shuts down and it never really happens, right? Because it never really gained the momentum to go full scale um, to help out the attack. However, if the infection persists beyond 4 hours, so let's say the blue line was not successful and you still have a lot of bacteria, that's when your induced response, right, kind of really takes over and is at an elevated state. So this is when you have things like big time recruitment of neutrophils and other physiological changes that we'll talk about today. The important thing to keep in mind is that after a few days, hopefully that's enough to beat the infection and the induced response fades away. But I want you to notice this. While this is happening, right, there still is a blue dotted line as well. Why? This is a reminder that the macrophages, the complement proteins that are part of this blue dotted line, part of your immediate response, they're still working. They're in the background still working, right? They don't just suddenly stop working just because your neutrophils have arrived, right? It is the combined effort of the immediate effectors and the induced response that succeeds in squashing the infection after a few days. Okay. So again, it's not really like an on and off switch. It's waves of activity and whether those waves have gotten high enough to actually encourage the next wave of things, of different physiological events to happen. All right. We had discussed the importance of cytokines as being what are in charge of actually starting up this induced response in the background as macrophages and complement C3 are doing their immediate work. We looked at this slide last time, and we said that many of the cytokines that are expressed by macrophages in response to detecting bacteria, right, either by eating bacteria, detecting it that way, or by having different receptor proteins on the surface of the macrophage that can pick up on the presence or evidence of bacteria. For example, the toll-like receptors on the surface of the macrophage that can pick up on lipopolysaccharide molecules that are shed by the kind of hairy dog bacteria, right, that have the thick lipopolysaccharide shag carpeting kind of coating. Right? We said either way, the macrophage starts putting out these cytokines, and some of these cytokines really stimulate inflammation. And we call these the interleukins. Right? Last time we looked at CXCL8 as one of the interleukins. It's also called IL8, right? It's another name for it. CXCL8 is one of the important ones that helps recruit different types of induced immune cells like neutrophils to come and actually find their way to the site of infection and start helping out with the macrophages. Let's look at another one today. This is IL-6. So IL-6, as you can see here, induces fat and muscle cells to metabolize. In other words, it increases the metabolic rate of your muscle cells, fat cells, and this is what increases your body temperature. You're raising your body temperature to hopefully raise the environment of your tissues out of the comfort zone of the bacteria. So the bacteria that's happiest at 37 degrees Celsius or 98.6 Fahrenheit, our normal body temperature, right? That's where they function the best. If you can at least raise your tissue temperature a couple degrees higher, maybe 99, maybe 100, that can slow down the activity of the bacteria, right? As a way for your immune system to kind of try to gain the upper hand and eat up all the bacteria, stop them from spreading. The IL-6 also has another important function, right? And that has to do with changing the expression of certain proteins that are made by your liver. So your liver, your hepatocytes are your liver cells, are of course normally making all kinds of things that are part of your normal liver function, right? Your liver kind of can detoxify certain things. Your liver is also a central place of how you regulate your overall body metabolism by managing things like glycogen and the amount of blood sugar that you have. But your liver hepatocytes are also capable of responding to the induced immune call for help by undergoing a process that we call the acute phase response. In other words, this is a very acute or very specialized, right, and limited response that your liver cells do, and in this case, as a way of dealing with infection. So your hepatocytes begin to express around 30 different special plasma proteins that get released into your blood, and these acute phase proteins contribute as part of your induced phase response against the bacteria or the virus. So a couple of examples are mannose binding lectin and C-reactive protein. Those are the ones shown here. Here's mannose binding lectin and C-reactive protein. There are other acute phase proteins as well, like fibrinogen, but we won't talk about those, right? Because I just want to get the concept across about how these are going to help your innate immune system. So let's start off with the mannose binding lectin, or MBL. So I already mentioned to you that lectins are a big group of proteins that have in common the fact that they bind to sugars of some kind, right? So we had previously looked at lectin receptors, and how if you have those special types of proteins on the surfaces of macrophages, that can help a macrophage recognize that there's a bacteria that's pressed up against it. Right? So this is an example of a soluble protein, a protein that's not really part of a membrane like a receptor, but it's actually made by our liver hepatocyte cells as part of this acute phase response, and it floats around in your blood and it starts flowing through your circulatory system. And the sugar that it binds is mannose, right? Because we already said evolutionarily, mannose is a very good target on the surfaces of bacteria because our body cells don't have nearly as much mannose sugars on their surface compared to something like a bacteria. So evolutionarily, it makes sense that we would learn to recognize that, right, as a general feature, right? So this mannose binding lectin, it's a very unusual shape. It's actually a triple helical structure that has sort of two parts to it. This long kind of twisted helical part, kind of like a stem, and then a globular part at the end. Right? Again, this is a nice reminder that these cartoons kind of grossly simplify the structures of things. But you know, this is kind of what it looks like, right? So you have 6 of these triple helices bundled together, and that means you have 6 of these triple mannose binding sites. So each one of these blobs at the end is the right shape to bind 3 different mannose sugars. So that means you have a total of 18 mannose sugar binding sites on this. It kind of looks like an inverted bouquet of flowers, right? In fact, if you look at these images down here, these are actual super high resolution electron microscopy images. You can see, right? It really does look like a bouquet of flowers, sort of inverted though. The shape of this inverted bouquet, right, is the right shape to also contain this other set of proteins, these MASP proteases. So these are other proteins that are proteases, meaning that they have an active site that can cleave or cut other proteins. Right? So they are positioned in here so that they don't cleave and they don't cut the mannose binding lectin. They're just carried around by it. So here you can see the MBL, the mannose binding lectin, has found the surface of a pathogen that has lots of mannose on it. So these red things are the mannose sugars. Right? And so the bouquet now attaches to the surface. Right? And because it carries these MASP proteins together, it's the right shape for a different complement protein, complement C4. Right? C4 is very similar in structure to C3. That's why they also show it as this like egg, but now instead of being a blue egg, it's like this pink egg, right? And C4 then, it's the right shape, as you can see represented here, to attach to this MBL. And the MASP proteins then, they're in the right position to go ahead and cleave the C4 into C4A, the little anaphylatoxin part that floats off and acts as like a little signal, right? And the C4B part that gets stuck to the surface of the pathogen. Right? Remember, this is happening because the original ability of C3 complement proteins, for whatever reason, are not doing a good job of getting planted on the surface of the pathogen as C3b. Right? Because that didn't happen too well, the macrophages maybe weren't having a good time eating the pathogen. Right? So over time, there's more and more pathogen. The macrophages have sent out cytokine messages in the form of things like IL-6 that have caused your liver to kind of wake up and start making things like this upside down bouquet, the MBL, right? And the MBL now can go ahead and attach, and it's recruiting this other complement C4b, and C4b gets stuck to the surface. Okay. Another acute phase liver protein that's made is this C-reactive protein. So when the liver does wake up in this acute phase response, some of these proteins like the C-reactive protein or even like the mannose-binding lectin, they get bumped up in production like 100-fold greater than normally what your liver cells are doing. All right, so it's really in response to this infection. Here you can see this is the C-reactive protein down here, this purple kind of pentagon shape, and you can see it's made of 5 subunits that together, right, make up the C-reactive protein. And the C-reactive protein sticks very well to another molecular component, phosphorylcholine, on the surface of the pathogen. Right? And when this sort of pentagon-shaped platform gets stuck to the surface because of that, it is a convenient attachment site for this other complement protein, complement C1, which looks a lot like that mannose-binding lectin that I showed you on the previous slide. Right? And I know as an immunology student, this kind of stuff can drive you nuts because you're like, I don't get it. How come one upside down bouquet is the mannose-binding lectin and this one's also an upside down bouquet looking thing, but we're calling this complement C1? You know, right? These proteins, they were not discovered at the same time. They were discovered by different research groups, oftentimes decades apart. And when these researchers are discovering and describing these proteins, right, they just give it a name that sort of fits with their impression of their function. So in this particular case, you know, this particular protein was sort of thought of as a complement protein. So it's called complement C1. On the previous slide, you know, that upside down bouquet was first studied because it binds to sugars, binds to mannose. So they just decided to call that a mannose binding lectin. But not surprisingly, I mean, they're all kind of related, right? Because they're all proteins coded for by genes that are part of our built-in immune response. So over millennia of evolution, it is not surprising that these genes are all related. And if you go far enough back in evolutionary history, they probably all started off from 1 or 2, you know, ancestral genes that over evolutionary time kind of got copied, put on different chromosomes, you know, over evolutionary time got mutations to slightly change the shape of the proteins that are made. So anyhow, complement protein C1 fits very nicely on top of the C-reactive protein. So if that C-reactive protein is now stuck on the surface, these are like little landing pads for the complement C1. And the complement C1 is the right shape, again, to recruit complement C4, just like the mannose-binding lectin does, and it cleaves the C4. And this is another way that the C4b gets stuck to the surface. Okay. So let's go back to this slide. This slide was probably the very first one that I showed you a couple lectures ago, introducing complement proteins. Right? We said that there are 3 ways or 3 pathways of complement activation that are part of our immune response. We started off studying the alternative pathway because the alternative pathway is the first one to act. Right? This has to do with the complement C3 that's just already floating around inside of your blood. Right? At this moment, you've got a lot of C3 floating around in your blood, right? Just to be ready in case you do get poked or cut and bacteria or other germs get in there. And we said that the C3 is activated, forms this C3 convertase, right? That then activates or cuts more complement C3 to help spread the attachment of C3b on the surface. Right? So today we've introduced these 2 other pathways, the lectin pathway, right, which involves the mannose-binding lectin that's made suddenly in large amounts by your liver, right? Your liver suddenly kicks in and produces like 100 times as much of this as you normally would have. So right now you have very low levels of this, relatively speaking, in your blood. But if need be, your liver can make a lot of it. So it's the second to act. right? So that mannose binding lectin will bind to the mannose sugars on the surface of the pathogen, resulting in complement C4b activation and attachment, right? Then we also said your liver will produce C-reactive protein. C-reactive protein that attaches to the surface of the bacteria like little platforms, and that's where the complement C1 will come in. And ultimately, that complement C1 also activates C4, making C4b. Once those C4bs are attached to the surface, they recruit complement protein C2. So C2 in this particular pathway is very similar in function to the factor B in the alternative pathway. And now you've created a convertase complex. Ultimately, this convertase complex, what is it going to convert? It's actually not going to convert more C4. It is the right shape to convert more C3, right? So the reality is all three of these pathways, even though they go through different intermediate molecules, is all about increasing the amount of C3 activation and getting more C3b onto the surface of the cell, right? Because that's sort of the most basic, quickest way that your immune system can help identify and label bacteria or viruses for destruction is by coating them with the C3b. Right? I want you to take a look at the fact that because both of these pathways, the lectin and the classical, right, require many intermediate events to happen. For example, the release of cytokines from the immediate macrophages that responded, and over several hours and into days, those cytokines have built up in order to do things like turn on your liver to make this acute phase liver response and make these other proteins. There's a delay. Right? So the lectin pathway is the second to act, right? And then the classical pathway is the third to act. I also want to point out to you that not only does the C-reactive protein that's made from the liver help out with this classical pathway, right? But there's another important type of protein that can also help out with the classical pathway, and those are antibodies. So antibodies can also attach to the surface of a bacteria if you have the right antibodies that recognize that particular bacteria. But if they do, they can also help with the recruitment of complement proteins to lead down this pathway, right? They get C4b onto the surface. That forms a convertase complex with C2a, and it's a C3 convertase that's ultimately going to convert more C3 and get C3b on there. So you might say, wait a minute, I thought antibodies were part of our adaptive immune system. Right, the whole thing, Dr. Shen always makes a big deal, right, about how our adaptive system is kind of put off. We try to avoid the adaptive immune response if we need it because it's expensive. That's the SWAT team, blah, blah, blah. And it takes a big toll on our body's resources. But antibodies come from that, right? Yes, I don't want you to be confused by this now, but let's just say, right, that antibodies are a product of you previously having to rely upon the SWAT team and the adaptive immune response to beat the infection by this particular pathogen, right? And it's because your adaptive response in the past was successful in saving your life, right? You now have antibodies that can then participate in a faster response as part of your innate response, right? In this type of a situation here. Okay, but don't worry, we will learn about antibodies in the next couple lectures. All right, so another important example of an induced response of your immune system, in other words, something that's not immediate, but takes time because there are some intermediate processes that happen, are how our bodies respond to viruses. Up until now, we've conveniently just looked at examples of pathogens as being things that are extracellular, meaning something like a bacteria or something like a virus that virus that comes into your body, and before it actually gets into one of your cells, right, the virus or the bacteria can be eliminated by things like macrophages and complement proteins, right? Because just as long as they are outside of your cells, things like the macrophages or neutrophils or complement proteins can get to them. Right? But what if something like a virus has already made it inside your cells? Right? Once inside the cell, macrophages can't get to them, complement proteins cannot get to them. So what does that mean? Does that mean that your immune system just says, all right, forget those, right? We were too slow, so now we just have to kind of suck it up and deal with the virus, like making us sick? No, obviously not. Obviously, we had to evolve a way to deal with things like viruses that are inside of our cells. In other words, viruses represent an internal cellular infection. Right? So how does our innate immune response deal with this? So here is a illustration. This is a typical flu virus particle that has genomic information in the form of DNA or RNA, depending on what kind of a virus you're talking about. There's some proteins in there, and it's surrounded by this capsule, right? So typical lipid bilayer, and you have these different protein spikes on the outside that allow it to gain entry into a host cell in your body. Right. So here's an illustration down here. This is an image of some red blood cells. Now, these red blood cells, I think you hopefully realize, mature red blood cells don't even have a nucleus anymore. Right. But the point is, the surface of the red blood cell still makes many of the proteins that the little green glycoprotein spikes of the virus particle can attach to. So these virus particles are trying to infect, and they are infecting these red blood cells, but, joke's on them, right? Because the red blood cell is not going to make more virus particles. But this photograph, I think, is pretty stunning in the sense that it's a reminder that it's not like one virus particle infects one cell, right? Here you can see hundreds of virus particles on the surface of these red blood cells, each one of which are trying to hijack the cell and make it do its bidding. On the right here, we see, so here's a viral particle. Its glycoprotein spikes are the right shape to bind to certain three-dimensional shapes of proteins on the outside of our cell. That's what makes it stick and allows the membranes to fuse. Inside the cell, the genetic material from the virus is released. And in this case, you can see the genetic material then is used by the host cell to make more mRNAs that are the viral mRNAs that are then translated by our cell's ribosomes into the finished proteins. They go through the Golgi complex where they're packaged up, and they appear, right, as part now of our cells. And then in this case, you can see these different components of viral proteins are able to then be assembled together to make a new viral particle, right? So clearly our immune system cannot afford to ignore that this particular cell in our body has been hijacked, right? Even though the actual pathogen is inside the cell, right? So how does our immune system deal with that? There's a very specific group of cytokines, small intercellular messages, that we call interferons. So interferons are cytokines that are expressed by our body cells when they are experiencing infection by viruses. So this is a way for one of our body cells that's taken over to actually call for help and let the immune system know that it's in big trouble. So these special cytokines that are made pretty much by all different cell types in our body in response to viral infection, and also natural killer cells that we will look at in a couple slides as members of our innate immune system. Natural killer cells also put out certain types of interferons to help coordinate the activity of macrophages, get macrophages involved in fighting these infected cells. There's two main types of interferons, two main families. Type 1, which is the type that's made when a cell is infected by a virus. Right? And so this infected cell in our body starts making these type 1 interferons. These include interferon beta and interferon alpha. Here down below, these are the type 1 interferons, interferon alpha and beta. And you can see, just like other cytokines, right? These are just small little proteins that are kind of folded up into their particular shape. Type 2 interferons are more specific. They're made by the natural killer cells in order to stimulate macrophages to work better or to induce inflammation. And there's only one form of type 2 interferon that we know about, and that's interferon gamma. So here's interferon gamma. You can see the shape is quite a bit different than the type 1 interferons, alpha and beta. Right? And here is the space filling models of these. So here's alpha, here's beta, and you can clearly see that gamma is quite a bit different than alpha and beta. But all of these are small little proteins that are the right shape to be used as signaling molecules, right? Because on the receiving end of these, whatever cell is going to receive these, there has to be a protein, an interferon receptor protein that is the right shape for these molecules to float around and bind to trigger something inside of the cell that has the interferon receptors on its surface. So how does this work? Let us say, for example, that we have a virus particle here that has infected this cell. This is a neat image because this is a virus particle that is fusing with the membrane of one of the body cells, and it's about to inject the genomic material into the cell. Right? So here it is over here as a cartoon. Right? It binds to the surface because it has certain glycoprotein spikes, these little blue triangles here that are the right shape to happen to bind to other proteins that are on the surface of our unsuspecting body cell here, right? So in this case, whatever this gray thing was, this is just some protein on the surface of our cells, and these blue triangles fit into it very nicely. So that's how this virus particle fuses with our membrane, our cell membrane, and inserts its genetic material. So it could be that this is double-stranded DNA if the virus carries around its genome as DNA. Right? Our body cells have evolved to not like the presence of double-stranded DNA in the cytoplasm. Because if you think about it and how a body cell normally works, our DNA is contained in the nucleus. Right? That's the only place our DNA should be. There should never be double-stranded DNA floating around inside of our cytoplasm. Right? Okay. Again, if that's true, and that's been true since the beginning of time, right, then it makes sense that we would have evolved innate mechanisms of detecting things like free-floating naked double-stranded DNA in our cytoplasm as being something that's wrong, right, and probably a sign of infection. Likewise, many of the viruses carry their genomic material as RNA, as double-stranded RNA. And again, our body cells don't like the presence of double-stranded RNA, right? Because most of the time, the RNA that's released by the nuclei of our cells into the cytoplasm is single-stranded mRNA that's waiting to be translated by ribosomes, right? So again, right, the presence of double-stranded RNA in our cytoplasm, we've evolved to detect as being something wrong. So this is how we detect it. Here we have, in this case, double-stranded viral RNA that's in our cytoplasm now. We have proteins such as RIG1 and MDA5. Okay, so these are called RIG-like receptors, or RLRs. These proteins are the right shape to bind to things like double-stranded RNA. So here you can see in the background is the big yellow double-stranded RNA, and these RIG1 proteins are the right shape to bind to it, right? These RIG1s are also the right shape to bind to these special RIG1 cards, these special protein domains here that are part of this large gray protein complex that's found on the surface of mitochondria. So this is where it gets a little weird, right? Because what does the mitochondria have to do with this? I would not have thought that our mitochondria actually helps out with fighting viruses, right? But it does. Right? We have these large mitochondrial mitochondrial antiviral signaling proteins, or MAVs, right, that have the right shape on their ends to bind to these RIG1 proteins. And these RIG1 proteins became the right shape because they started binding to double-stranded RNA, and they would attach there and associate with this MAVs protein. Okay. The reason why, if you think about this, it's actually kind of neat to imagine why the mitochondria is involved in this. Way back in evolutionary history, right, as eukaryotic cells were first developing, life on Earth at that point were just things like prokaryotic cells, like simple bacteria. Simple bacteria also have to have defenses against things like viruses, right? Because bacteria are also subjected to attack by viruses. For example, phage is the type of, or a group of viruses that attack bacteria. So these bacteria must have had mechanisms for identifying the presence of viral genomic material. Right? And when those prokaryotes, those prokaryotic cells, the bacteria, became part of eukaryotic cells, because remember there is this theory that that prokaryotic cells developed a symbiotic relationship with early eukaryotic cells because the bacteria could produce energy, produce ATP, right, that our eukaryotic cell could benefit from. And in exchange, the eukaryotic cell that surrounded them, right, provided them protection, source of nutrients, things like that. So over evolutionary time, primitive prokaryotics became the mitochondria of our cells. And that's why our mitochondria to this day still carries separate genetic information in it compared to the nucleus of our main cell. And that's why, right, the mitochondria still retain the ability to detect viruses. I think it's kind of neat, right? And that's how we evolved, because our mitochondria help us detect the presence of viral genomes. Okay, so what happens next? In this cartoon here, let's say you have double-stranded RNA from the virus, right? It's detected in our cytoplasm by RIG1s or MDA5s. They're very similar. These proteins bring the RNA to the MAVS proteins that are on the surface of the mitochondria, right? Because again, by binding the double-stranded RNAs, the MDA5 and the RIG, their shape changes. So now it's able to attach to the MAVS. Okay. On the MAVS protein, there's a protein known as IRF3, or interferon regulatory factor 3. When the RIG and the MDA5 arrive, that results in the phosphorylation of IRF3, meaning a phosphate group gets put on it. And that, of course, is going to change the shape of the IRF3. It falls off the mitochondria, and it goes and it finds this transcription factor, NF-kappa B, right? We looked at NF-kappa B before. We said that NF-kappa B is a very important transcription factor that's in charge of turning on many types of cytokine genes inside the nucleus of cells. So sure enough, the activation of NF-kappa B by IRF3 activates NF-kappa B, and NF-kappa B goes into the nucleus, and it sits on the regulatory regions like promoters or enhancers of interferon genes, right? And as a result, the production of interferon proteins happens. And these interferon proteins then can then be released by the cell as a cytokine message that the cell is infected by virus. So this is what we call the interferon response. Right? So these interferon cytokines, the name interferon, I like the name because they were originally identified as being small cytokine messages that interfere with viral infection, right? They're a way to block the virus infection from spreading. So there are three main components to the interferon response. One is that the expression of these interferons actually changes the cell itself that's infected, right? And it begins to alter certain functions inside of the cell. For example, it'll slow down things like DNA replication. It'll slow down things like protein production, right? And those things will result in fewer virus particles being made. Interferons can also increase the expression of different types of ligands or small molecules that receptors on the surfaces of natural killer cells can recognize. And these interferons can activate these natural killer cells themselves and get them to come and actually kill the virus-infected cell. So in this cartoon here, you can see that this cell has been infected by a virus. So the virus, here's the genomic information. Maybe it's double-stranded RNA, for example, that will activate on the mitochondria, the IRF3s, that end up resulting in NF-kappa B, turning on the expression of different interferons, for example, interferon B. So you get these little triangles, these little blue triangles that represent the interferon B protein that's made, right? And they then are released by the cell. So you can imagine then that you would have a cloud of these little blue triangles emanating from this cell, right? On the surface of the cell, there are receptor proteins that are the right shape to receive the blue triangle. For example, these are type 1 interferon receptors. So some of the blue triangles in this cloud that's kind of coming from the cell end up feeding back and binding to the type 1 interferon receptors of the same cell, right? And that's what then stimulates the cell to change some of its behaviors to make the virus replication slow down. It also stimulates the production of other interferons like interferon alpha. So this kind of a feedback loop, we would say, is an autocrine loop. It is the cell signaling itself to undergo some changes. However, even more importantly, right, these interferons are received by the interferon receptors on neighboring cells. In this case, a cell that has not been infected yet. But it receives this message from the infected cell that warns it, hey, heads up, there's a virus infection going on. So this uninfected cell can also change its cell biology to make it tougher for virus particles to infect it. For example, it can change the protein makeup of the cell membrane so that maybe the virus particles have a harder time attaching to it and infecting it. Okay, so that brings us back to this cartoon that we come back to quite a bit. It's a nice way to kind of reset and appreciate where we are in our discussion. Right? So we've talked a lot about different innate effector cells, like monocytes that become macrophages. We've talked about neutrophils quite a bit. Let's finish up today talking about natural killer cells and dendritic cells. All right, these two groups here. Let's start with the NK cells first. So natural killer cells are large lymphocytes. They come from sort of the adaptive side of our immune system that are very quick to respond to viral infection. So their jobs are to destroy the infected cells and also increase inflammation. You can see here in response time after viral infection, if the yellow thing is the virus titer, that means how much virus you have in your body. Very quickly following the production of interferons, you'd have a big boost in NK cells moving in and actually killing the infected cells. So this happens well before your T cells or B cells of your adaptive system are even mobilized. How do the NK cells work? They work by forming synapses against a target cell. So in other words, here you have, let's say, an infected cell. These are natural killer cells. They will come up and press against the target cell. And in that junction, where the two cells are pressed up against, you'd have a lot of interactions between different proteins on the surface of one cell and receptors on the surface of the other cell. Right? If the NK cell receives signals that really make it realize with quotes, that this is an infected cell, then what will happen is the NK cell will send different cytotoxic messages into the cell, telling it to die. So that's what we see here. We have an NK cell pressed up against an infected cell. So in this little space between the two, it's sending cytotoxic messages into the cell. And within a relatively short time, that cell undergoes apoptosis, okay, and it dies. Okay. Why don't I stop there today? I have a couple more slides, but we can just start off our next lecture with those slides. So in the interest of time, let me just go ahead and stop.